All right, guys, today for our AP test prep review, we're going to go over cell communication, immune response, and viruses. What this is more of a higher level connecting what I talked about yesterday with cell membrane to a new topic. And so I think these are the three topics that really connect with cell membrane really well. So I am going to go over each one of these. You have a handout to fill out. As you notice, this handout is just mostly full of diagrams. And what I'd like for us to do is we're going to go over more of the basic information on cell communication, but when I start talking about immune response and viruses, I don't think memorization is quite as important as just practicing reading, looking at, analyzing those diagrams. So that's why I did the handout the way that I did it. And hopefully I can pause if I feel like maybe you should write something down next to that diagram. But really I'm showing you examples of how cell membrane plays a huge part in immune response and how viruses infect a cell. So this is just review. Everything I talk about today we've actually covered before. This is just a quick review of the three stages of cell signaling: reception, transduction, response. So reception is going to deal with a signaling molecule or a ligand coming in from outside the cell. Transduction is going to convert that signal to a form that can bring about a cellular response, and the response would be the response. And so I'm going to go over each one of these. So just kind of jog your memory a little bit. And so how does cell signaling relate to the cell membrane? Because we have our plasma membrane right here. And if we have a ligand that is hydrophilic or water soluble, like insulin or growth hormone, then it's going to need a receptor that's embedded within the cell membrane. And so you notice right here, this is our cell membrane. You notice that you have your receptor right here. It's purple, so you can automatically assume it's a protein, so it's made up of amino acids. And then you can also assume it's spanning the entire membrane right here. So it must be an amphipathic protein, meaning it has hydrophobic interior amino acids and it has hydrophilic amino acids on the outside. So that's why I'm relating this topic to cell membrane. So the signaling molecule comes in and it fits very perfectly. The basis of many diseases or genetic diseases is going to be a DNA mutation that causes a change in amino acids that affect the folding of a receptor protein so that a ligand can no longer bind to it. So that's why this is an important topic. Transduction is going to deal with a relay of molecules, conformational changes of proteins that propagate a signal throughout the cell. Because remember, if this ligand is hydrophilic, it cannot enter the cell itself. So there's something that it will have to cause, like a domino effect, that passes through the cell all the way to probably an enzyme or the nucleus or something like that to give a response. All right, so let's talk a little bit about the reception part of this. I just went ahead and copy and pasted this onto your paper so that you didn't have to write. Like I said, the binding or the fit between the signal molecule and the receptor is actually highly specific. And we have two types of receptors. We have receptors that are embedded within the plasma membrane. Those are going to bind your hydrophilic or water-soluble ligands, like insulin. And then we also have intracellular receptors. So intra means inside, cellular, the cell. So we have receptors that are actually inside the cell. So what kind of ligands are those going to bind? Well, they're going to bind hydrophobic or small ligands that basically really can just pass through the cell membrane freely. They don't need a receptor within the membrane. The ligand's going to bind to the receptor changes shape. So another way of saying change shape is conformational change. I would definitely remember that. And then that initiates a transduction signal. These are the three types of plasma membrane receptors. So we know that if they're embedded within the plasma membrane, they are going to be specific for a certain hydrophilic ligand. And so if you remember, we talked about G protein coupled receptors, tyrosine kinase receptors, and ligand-gated ion channel receptors. I wouldn't necessarily have anything memorized about these What I would do though, when you're reviewing for the AP exam, if you see on the next page of your little note packet, I have all three types. Can you talk through what's happening in each one of these pictures? So it looks like here, everything is inactive. Then in step 2, a signaling molecule binds. Step 3, the G protein became activated, moves over, and it activates an enzyme, causing a cellular response. Then the G protein is dephosphorylated, or phosphate groups comes off, right? So it's just a series of steps that result in a cellular response. All right, receptor tyrosine kinase, what makes this one unique is that it requires 2 ligands. I bet you're all remembering this right now. 2 ligands that bind, and then it forms the 2 tyrosine kinase receptors come together to form a dimer. 6 phosphates get added to it. And what's really unique about this one, besides requiring 2 ligands, is that it can cause multiple cellular responses. So we said that receptor tyrosine kinases were really important in cancer. And in particular, we talked about breast cancer treatment. And the tyrosine kinase receptor that's involved in at least some forms of breast cancer is the HER2, the HER2 receptor. I find that kind of funny. It's not funny, but it's kind of like, that for breast cancer, it would be HER2 because mostly females are going to get it. The drug that's used to treat some forms of breast cancer is called Herceptin. And what Herceptin does, so if a HER2 receptor is overactive, that means too much growth hormone or whatever the signaling molecule is binding to it, causing too much growth and cell division. So what Herceptin, the drug that's going to block the HER2 receptors, does is it blocks right here so that the signaling molecules can never bind, and so the signal for the cells to divide never occurs. I like that name Herceptin because it's the HER2 receptor, and it's like I guess I think of it as like an interception. I always think naming drugs would be a really fun job to have. I'm not sure how to get that job, but I would really love that job. All right, and then ligand-gated ion channels. This is the easiest one. This is my favorite one. Signaling molecule binds, gate opens, ions go through, gate closes. This is going to be important in things like sending a cellular response when a person is experiencing a pain stimulus. So when you go to the dentist and the dentist sticks a needle in your mouth, that is going to open sodium channels and a signal is going to get sent. Push the dentist away. So that's the reason why they're going to give you lidocaine shots in your mouth, is because what lidocaine will do is it will block those sodium channels so that the pain signaling molecule cannot bind. And so the signal never gets sent to your brain that someone is like cutting into your gums. So lidocaine is such a cool drug, I think, especially because it wears off. I mean, it just lasts the perfect amount of time to do dental procedures. It's pretty cool. All right, so those are just the 3 types of receptors. Proteins are important in that. The fit between ligand and the receptor is important. And the other thing is just conformational changes are going to result. That's part of the domino effect. So another or the next step is going to be transduction. We've heard of transduction before, and this is really confusing because we use the word transduction to describe when a bacteriophage virus infects a bacterial cell and basically not rearranges, but inserts its viral DNA into the host genome, therefore changing the host genome. That is called transduction. So I wouldn't forget that. But the transduction for this section actually has to do with just the propagation of the signal. So remember, it's a cascade. The protein kinases are the enzymes that are involved, and you have this phosphorylation cascade, a series of phosphorylations and conformational change that propagate a signal all the way to the place in the cell where the signal needs to cause a response. Okay, and that's really it for transduction. Hopefully you got this right here. You filled in those blanks right there. The protein kinases. Protein kinases are really important in the treatment of disease too. So not only can a disease be caused by a faulty receptor, it can be caused by faulty protein kinases. All right, and then last we have a response. And our response is going to either be turning genes on and off, activating enzymes, or anything like that. So this is a pretty good overview of reception, transduction, and then we're inside of the nucleus here, response. And so in this picture, it looks like the response is the initiation of transcription. So that mRNA gets made. Most likely it's the mRNA that is responsible for a certain enzyme. That mRNA is going to then leave the nucleus through the nuclear pore, get translated into a protein, and now you have that protein that's going to carry out the task that this original signaling molecule wanted to happen in the first place. Most likely with this scenario right here, you see the signaling molecule's growth factor. Growth factor is responsible for cell growth and division. Most likely the protein that's going to get made here is a protein that's involved in the cell cycle and giving that green light to proceed to mitosis. So if you guys can kind of remember, we had that wheel and you had G1, S, G2, M, and then you had these CDKs, cyclin-dependent kinases, and then cyclins. Those are probably proteins that are going to get expressed when a growth factor binds to a receptor of a cell. So it just kind of puts everything together, right? All right, I'm going to move on to the immune system as an example of cell communication. Once again, I would not memorize everything. I would just look at these diagrams like, lots of receptors involved here. Wow, lots of cell communication involved here. Pretty cool stuff. So I talked about innate immunity. Innate immunity is your first line of defense. These are all the things that are going to happen when the body senses that there is a foreign invader. Interferons, these are antimicrobial proteins that are going to get released that inhibit viral reproduction. Natural killer cells are going to kill cells that have been infected with viruses and also cancer cells. White blood cells, in particular your macrophages, are going to be activated and they're just basically going to eat the foreign invader, digest it, and remember what they do is they present a piece of the foreign invader to T cells that's going to kick off adaptive immunity, that next phase of immunity. And then inflammatory response, which is probably the quickest, most immediate response, is that mast cells are going to release histamine, blood vessels dilate, redness, swelling, itching, fever falls under inflammatory response. All of that is to increase blood flow to the area of infection, which increases all of these other things to the area of infection. And then fever obviously is going to raise the temperature in hopes of denaturing the proteins of the foreign invader. And then I didn't mention barrier defenses. Your skin, your mucous membranes, and then enzymes in your tears and saliva and mucus are going to be involved in innate immunity. And sorry, my presentation got a little bit messed up here. Let me run through this. Apologize for that. All right, so I just want to give you an example of receptors. Receptors that are located on the plasma membrane of phagocytic cells, like your macrophages, are going to activate a response. So they're called toll-like receptors. And you see right here, toll-like receptors are going to bind to any parts of a foreign invader. So you see a lipopolysaccharide here, flagellin right here. It looks like there's parts of an invader, and what that's going to do is it's going to signal a response that's going to result in phagocytosis. A little bit about the inflammatory response. Mast cells and macrophages are both going to release signaling molecules that are going to basically recruit neutrophils, which is another type of large white blood cell that's going to help and kill the foreign invader. And so just an example of, all I really want you to know here is just an example of signaling molecules get sent to recruit other players in the immune response. All right. Innate immunity is not enough though to, for us at least, for large eukaryotic multicellular organisms, it's not enough. So what we have is a second wave of events, I guess, if you will, that is called the adaptive response. And so if you remember, it can take several days for the adaptive response to really fully kick in. And so what we have, the players that are involved in the adaptive response are T cells and B cells. T cells, there's 3 types: helper, cytotoxic, and memory. And I don't know if you guys remember us drawing that diagram with all the arrows, just showing like the domino effect and the chain of events that leads to activation of all the different white blood cells. So what helper T cells are going to do is recruit other T cells, B cells, and phagocytes. Cytotoxic T cells are going to kill infected host cells. And then you have your memory T cells, which are going to be stored upon subsequent exposure. All right. So what happens is during innate immunity, macrophages are going to engulf a foreign invader. They're going to chop them up. So if you can remember me standing at my demo table and I'm tearing up the pieces of paper, and then I have the one little piece of paper that I'm basically bring out to the cell surface, and then so that's my antigen, a little piece of a foreign invader that's going to be presented to a T cell, and that's what's going to kick off adaptive response. And so I wanted to show you this diagram here because T cells have specific receptors that are going to fit that antigen perfectly. All right? And so that's how it will know this is the foreign invader we're going after. So the way that I think of this, and I have like an example, or I have an analogy that I think fits the situation. It's the best one I can come up with. I think someone has said something about, oh, they're like the drug dogs. And I'm like, yes. So let's say you have like, it's New Year's Eve and you have like a DWI checkpoint or something like that. The checkpoint would mean that every single car is getting checked. And if anyone shows any signs of inebriation, I guess, or they're not able to drive, or they're not able to talk right, they're going to get a field sobriety test and get thrown in jail, no matter what the substance is that they're using. That's like a checkpoint. So that would be your innate immunity. Checking anything that comes through, anything that seems off, just throw them out, basically. What your adaptive immune response could be, the analogy is at the same checkpoint, you have drug dogs that have been trained to locate specific drugs. So let's say they're doing this checkpoint, anyone that goes through that shows any type of impairment gets thrown in jail, but they're also trying to target they're trying to figure out if there's some cartel members driving through. So let's say methamphetamine is a big problem at the border of Texas. I don't even know if it is. I know cocaine is, but methamphetamine's been flooding in across the border. So we have this DWI checkpoint, but we also have these drug dogs that are trying to locate the methamphetamine. Hopefully that made sense to everyone. They have been specifically trained, if you know how a drug dog is trained, they've been trained to sniff out specific drugs. So while innate immunity is non-specific, anything that seems off gets thrown out. It is not enough if there's a particular type of pathogen that is overwhelmingly invading the body. So this is where adaptive immune response is going to come in. They are going to be specifically fit to only that pathogen, and they're going to launch an attack on only that pathogen. So hopefully that made sense. What this shows you right here is your macrophage presenting an antigen to a T cell. Cool. And I wouldn't pay much attention to this right here. You have actually we will. So your macrophage is presenting an antigen to a T cell. That T cell then becomes a helper T cell, basically sending out signals like, hey, other T cells, I got something we got to be looking for. Hey, B cells, we need you to start making some antibodies against this. And then another type of T cell that's going to be activated are your cytotoxic T cells. And what they are going to do is they're going to hunt for host cells that have been infected by the pathogen, and they're going to signal the host cell to undergo apoptosis. All right? And so now we have our B cells. So your helper T cell has been tagged, like right here. You have your macrophage, that's a part of the innate response, has a little piece of an antigen like, hey, this is what you're looking for, this fits you perfectly, you need to rally all of your other T cells, start dividing, and start rallying the B cells to start making antibodies. So what B cells are going to do is they're going to release antibodies. Antibodies, and the red thing in this picture right here. Antibodies are going to also have a very specific fit for the pathogen, have a very specific fit for the antigen. So they're not just going to surround anything, it's a very specific fit. So there's two types of B cells, plasma B cells and memory B cells. Plasma B cells are going to release antibodies. Memory B cells are going to be stored for subsequent exposures. So what do antibodies do? Three things. Neutralization, so they surround the pathogen, rendering it inactive. Opsonization, which means it's going to tag it for destruction by phagocytes. And then antibodies are going to activate a complement, which enhances opsonization and lyses some bacteria. So the complement activation, we didn't really go into that very much, but these two are the most important. Neutralization, opsonization. Surround them, rendering them inactive, or tag them for destruction. So antibodies are very, very important. And we know that this is really like the climax of an immune response, because if a person has HIV, this right here doesn't work. So what do helper T cells do? They recruit other T cells. They actually recruit B cells. And we all know what happens when a person with HIV or AIDS, what happens to them. They end up with opportunistic infections that you would never, any just normal person walking around wouldn't have that wasn't infected with that virus. So antibodies are so very important. So those are the two things that antibodies do. All right, so this leads me in, and then that's immune response in a nutshell. You have your innate immune response, and the innate immune response gets connected to adaptive immune response with this antigen-presenting cell. The antigen-presenting cell is going to tag the helper T cell. Helper T cell tags these guys. Cytotoxic T cells and kill infected host cells. B cells are going to release antibodies. So that's it in a nutshell. So I want to talk about viruses. I'm going to talk about coronavirus a little bit here, because that is kind of relevant right now. It's the reason why you're watching this instead of actually sitting in the classroom today. So the first thing that is really important about viruses is they have to have some kind of entryway into the host cell. So here's the host cell, here's the virus, and right here I have influenza, which is the flu virus. I'm going to show you a picture of coronavirus here in just a minute. They're kind of similar, at least in the way they look. So what happens is that the virus has to have some kind of entryway, and the key, literally, is a protein that is on the surface of it. It has to somehow unlock the cell membrane so that the cell membrane will undergo endocytosis and take it in and form a vesicle around it. And so what happens after the cell basically engulfs the virus, just like, hey, come in, friend. The virus uncoats itself. And so we have proteins, RNA molecules. RNA will then enter into the nucleus, or if it's a DNA virus, it'll enter into the nucleus. This is RNA. The RNA is then used to make proteins, and voila, we have new virus that is then going to leave. So basically the virus brings its genetic material, hijacks the cell, making the cell produce more virus for it, and then it leaves to infect other cells. This is a really, really bad picture. This is the picture from your textbook and from the notes I'd given you before. It's just a little bit more detailed overview of what actually happens. So virus enters, RNA is used to make more RNA, more proteins, more glycoproteins, and then virus is made, and it uses exocytosis and the actual host membrane. So the new membrane that surrounds the virus is actually just host membrane. Right, so this is what coronavirus looks like, and so what researchers are trying to study are, what are these proteins? What are these proteins? What are these glycoproteins that are on the surface of this virus that is helping it gain entry into cells. And so those are being sequenced. Some of them, as I understand, have already been sequenced. Some are still to be sequenced. And so something that's really interesting, this is also called SARS-CoV-2. So this is coronavirus, COVID-19. It actually targets a receptor called the ACE2 receptor. And so it has been found that it has a protein that fits a receptor on the cells, the epithelial cells in the lungs, that is helping it gain entry. So it's like a key, a lock, it unlocks it and the cell brings it in. And so once it enters into the cell, I'm sure you all have looked this up. This is all over the place. But this is the innate immune response right here. Basically being launched right after infection. Now it can incubate for 1 to 14 days, which is a lot different than the flu and the cold. So that's kind of scary. And the symptoms are pretty gradual. If you've ever had the flu, I mean, you're fine 1 minute and then the next minute, I mean, it is like you got hit by a train. From what I understand, this one's pretty gradual. You just start with a little cough and then it gets worse. And then you get a fever, and then it gets worse, and almost like 5 days into it is when the respiratory distress really starts. And so here's just a symptoms list, and the reason why I'm showing you the symptoms list is normally when you have a symptom of a virus, that is your immune system trying to get rid of it. In particular, fever and cough is trying to get rid of it. All right, so when a virus basically attacks the lungs like coronavirus does, the lungs can't efficiently get rid of carbon dioxide. And we know when carbon dioxide is in the presence of water, it turns into carbonic acid. Carbonic acid is really great. It's a great buffer. But we have a very, very strict homeostasis process that makes sure there's not too much carbon dioxide in the bloodstream so that pH doesn't go down too low. So what this does is it causes an acidosis, a respiratory acidosis. The blood becomes too acidic because not only can you not get enough oxygen in, you can't get enough carbon dioxide out. And I think that's the part that some people don't think. If they're like, oh, I can't get enough oxygen, but what's really going to cause a lot of problems down the line is not being able to get rid of carbon dioxide out of the lungs. And so pH gets too low. And we know when pH gets too low, I mean, that's going to cause all sorts of problems. And the first thing you should think about when we talk about pH is, denaturation of proteins. So what happens when pH gets too low, the kidneys are going to compensate for this. And what the kidneys are going to try to do is carbon dioxide is going to combine with the hydrogens of water to make carbonic acid. The kidney is going to take that hydrogen back. Well, what really stinks about coronavirus is it is believed that not only does it attack the epithelial cells of the lungs, it is actually showing in many patients, not all, but many patients, that coronavirus is gaining entry into the cells of the kidneys. The kidneys are the ones that are trying to fix the problem that it's causing with the lungs, but this is like a double whammy because it is now taking the one organ that's trying to fix respiratory acidosis and it is infecting that organ too. So just evil, horrible. And so what a lot of patients are experiencing is called acute kidney injury. And so I'm not going to read word for word through this. You can pause it and read it if you want to. But it says that the mechanism is unclear. It is believed that there's a cytokine storm. Cytokines are signaling molecules that recruit more immune response. But then there's also a direct cellular injury due to the virus itself gaining entry into the kidney cells. And I'm not going to read through all of this. It's just a lot of fancy words. And all of this is really saying is that it is now believed that coronavirus can gain entry into kidney cells. So remember, viruses are tissue specific and host specific, but it looks like this one is a lot different than the flu because it can gain entry into kidney cells. So that's a really scary part about it. So kind of onto the brighter side, and then I'm almost done with this talk, is what do we do in this situation? So with any virus, using drugs, Normally using antibodies isn't something that is done, but in this case, with this type of crisis, they are trying to infuse patients with the antibodies of patients who have recovered from coronavirus, and then vaccine. So vaccine, that would be the ideal thing to do. I don't want to say treatment because vaccines are for when you haven't had it yet, but vaccines preventing the virus is the best thing to do. But we all know, and I'm sure you've all heard, that's going to be a while. They're not just going to start shooting people up with just anything. It's got to go through many, many steps and many, many processes. So the very first thing that doesn't take very much time is the repurposing of old drugs. And the one that's been in the media is one called hydroxychloroquine. Hydroxychloroquine is a drug that's used to treat malaria. Some way, somehow, hydroxychloroquine is actually a very basic drug. And so what it does is it changes the pH of the cell membrane. And you know when pH is changed, there's like a cascade of events where certain chemical reactions cannot happen. And also it causes denaturation of proteins. So what you have with hydroxychloroquine, it's only one of the many mechanisms that it does to prevent viral infection. It's believed to change the pH of the cell membrane so that when coronavirus tries to dock onto that receptor, because of that major pH change, it now can't not make its way into the cell. Now, I know there's a lot of political stuff behind hydroxychloroquine and certain people endorsing it who have a stake in the stock or as a shareholder, I don't even know. It is something worth looking at. From what I understand, though, some of the clinical trials have not been very favorable. I think they are still testing it, and there is some hope that it can do something. I think something is better than nothing at this point, but this one's gotten a lot of attention so far. There are some cancer drugs that have actually gotten some attention too, but just for the sake of time, I'm just going to go over this one. So verdict's still out on this drug, unfortunately. The other thing that's being done right now is like I said a minute ago, they're begging people who have recovered from the COVID-19 to please donate your plasma because plasma will have the antibodies that fit the receptors or the proteins on, not the receptors, that will fit the proteins on coronavirus really well, blocking it from ever making contact with that ACE2 receptor. And I didn't state the study here. You're just going to have to take my word for it. It has been found in many cases. I don't even know what the number is. It's a study where they actually only talked about 5 patients, but I know there's many studies out there. It reduces the duration of fever. It reduces the duration that a patient is on the ventilator, and it reduces the duration of ICU and overall hospital stay. This has been something that has been very favorable, but as you all probably know, it's not. I mean, it's a limited resource, and it has not a whole lot of research has been done on it. All right, so other thing I wanted to bring up is how do we know when someone has the virus? They're doing ELISA testing. I don't know if you guys remember, we had the scenario where all these different people came into contact with each other and someone had an HIV, and so we did this exact setup right here. So this is what is done when someone's getting tested for coronavirus is ELISA testing. They're testing for the antibodies. So ELISA testing tests not for the virus itself, but tests for the antibodies. So if you ever have the opportunity to get tested for coronavirus, this is what they're doing, and you have kind of a reference because we did it in class. And then last thing is it's important to just look back at what a vaccine actually does. So a vaccine is going to have little bits and pieces of a dead virus. It is going to launch a response, and it's going to take several days for antibodies to get made. So then a person has lots of memory B cells. Time goes on, secondary exposure. So when a person has the vaccine, they've already made the antibodies. A huge immune response hasn't been launched because of dead attenuated virus that's put into these vaccines. Then if they're ever exposed to it again, they have those memory cells that are going to launch the adaptive immune response, and they won't even know that they've been come into contact with the virus. So my goal was to keep this under 35 minutes. I hope you found it a little bit interesting. I really like talking about immune system and viruses. And I think While you don't need to memorize everything that I talked about, I do think making those connections between cell communication, immune system, and virus, and cell membrane is really important. All right, I will get you guys an FRQ practice tomorrow, and then we'll do a Zoom meeting on Friday. Talk to you later.